senses to cover today, the eye and the ear. And I'm going to do the ear first because I tend to talk about the eye too long and I don't have enough time left for the ear. So I'm going to move to the ear first and then we'll come back and finish up with the eye. And as most of you hopefully already know, we have our next exam on Wednesday, which is a little unusual for us. So you don't have the normal weekend between today's new material and Wednesday's material. Hopefully you've made adjustments for that over the last couple of weeks. Otherwise, the schedule will be the same. So we'll have our lecture exam in here at 7:30, and then a lab exam over in the lab room. A little bit of a difference over the last couple of years, Dr. Carter and I have added a few extra questions for the extra credit side of the exam. So instead of 72 questions, there will be 75. So 3 extra questions, but still divided by the same number. Okay? So just planning in your timing for the exam on Wednesday. All right, so when we talk about special senses, we're still talking about sensory neurons. For the 2 special senses that we're talking about for hearing and for the eye, we have cranial nerve. And anybody remember what the sensory relay area of the brain is? All of our thalamus, good. So all of our sensory neurons pass, except for smell, pass through the thalamus. This is gray matter, collection of cell bodies. No, that's why we use the term nuclei. So remember we had 2 neurons for the motor pathway, upper motor and lower motor. What structure in the brain is created by the gray matter formed of the cell bodies of the upper motor neurons? Can you be more specific as to the gyrus? You're getting there, yeah. Precentral gyrus, okay. And the gray matter in the spinal cord formed by the lower motor neuron cell bodies? No. Well, what did you say for the first part? Anterior, yes. Anterior gray horn is the cell bodies of the lower motor neurons. Okay. So for our sensory, how many neurons do we have in the pathway? 3. Yeah, first order, second order, and third order. So what structure is formed by the cell bodies of the first order neurons? Pseudo-unipolar neurons. The inside the central nervous system or outside the central nervous system? Outside. So we would call it a nucleus or a ganglion? A ganglion. So what do we call the ganglia associated with the spinal cord at least?
Dorsal root.
Dorsal root ganglion. All right. There are ganglia for our sensory neurons associated with the cranial nerves as well, but they each have their own name. All right. So we don't have specific names for the dorsal root ganglia, but for the cranial nerve sensory, the vagus and the trigeminal components would have their own. All right, where do we find most of our second order neurons for sensory? We're talking about spinal cord. Posterior gray horn. All right. Now, some second order neurons, a few of them aren't until the medulla, but we're not getting that specific into those pathways. And then the thalamus is formed by the third order neurons, the cell bodies of the third order neurons. Okay. All right, so we'll look at that in a little bit more detail when we look at the eye pathway. I just wanted to lay down a little bit of review material. So looking at the ear, we have 3 regions. So remember I said a few minutes ago we're going to skip ahead and do the ear first. So there are 3 regions. The external ear, middle ear, and the inner ear. The first 2 of these regions contain air within those spaces, right? And when we get to the inner ear, we have a series of structures that demonstrate fluid. What are the 2 functions of the ear? Okay, hearing and balance. So in our vestibulocochlear nerve, cranial nerve 8, we have those 2 functions. Remember I said on the lecture exam, when I name a cranial nerve, I will give you both the name and the number. Okay, but on the lab exam, you need to give me the name. So balance and hearing. We see those functions in the inner ear. external and middle ear have to do just with hearing. Okay. So our structure of the external ear is to capture sound waves. And we have the, well, not quite large enough to be an ear trumpet, but we have the pinna, or auricle of the ear. Remember on the sacrum and the ilium we had those auricular processes that looked a little bit ear flap shaped. And so this is identified as the auricle or the pinna. Now, we think that ears are most attractive when you can't see them. Okay? So if they're flat against the head versus at perpendicular angles. And so individuals that can afford, if they're sticking way out, and that's one of the characteristics that cartoonists draw of Obama's, you know, the big ears sticking out, always look for a chin or a nose or something that they can exaggerate. And so that really doesn't, when they're flat against the ear, or even if they stick out at right angles, it really doesn't work as well as some ears for capturing sound. So you'll see individuals that are hard of hearing that will put a, you know, a cup shape around their hand. And the first hearing aids were large trumpet-shaped structures that narrow down in an attempt to capture sound waves and direct them towards the hearing apparatus. So that's the role of the auricle or the pinna, is to capture those sound waves. And then they're directed into the bony opening that you are aware of as the external auditory meatus. But that becomes the continue calling it the external auditory meatus, or you could call it the external auditory canal. And all of this is lined with our skin epithelium, right? So cornified, stratified squamous epithelium. As we get to the confines of the middle ear, the last structure that belongs to the external ear is the primary tympanic membrane. Now, I believe your lab manual just refers to it as the tympanic membrane. There are actually two. And so our layman's term for this is just the eardrum. Notice that it's not a flat disc. Okay? It's tilted at an angle and it is a slightly depressed. And there are individual variations to the shape of the eardrum. It can be, instead of being depressed and disc-shaped, it may be really flat, or it may be inverted towards the auricle. That affects the pressure within the middle ear. And when we get to the internal auditory tube, I'll talk about that a bit more. Now, this external auditory canal is now passing through bony tissue. Right? So we're going to have the temporal bone up here. And the middle ear, continuing from the external auditory meatus, is a space, or a room, if you will, within the temporal bone. So we continue to see a bony component to this, and then our internal auditory canal is bone and then cartilage. So as this leads to the back of the throat, it becomes cartilage. So our middle ear, like the external ear, is another air-filled space. And it's going to continue to be lined by a mucous membrane, which is going to be continuous with the internal auditory canal. This would be a non-cornified, secretes a serous fluid. Okay, but the space itself is air-filled, so it would have a moist surface. Now, if the role of the external auditory ear structure is to corral the sound waves and to direct them towards the inner ear, what would be the role of the structures of the middle ear? Most of you are familiar with the structures of the middle ear. These are three small ear bones or ossicles. Right? So we're going to see these ossicles. There are three, two of which are attached to a membrane. The other one is found in between. So when you look in the lab, you will see a long bone attached to the deep surface. This is a little bit larger. Attached to the deep surface of the tympanic membrane. And this one is known, I'm just going to put a number here. It's going to be in my way. I'll move that. As the malleus. And if we translate that, it means hammer, like a mallet. Okay, attached to that is another bone that kind of crosses this space. And that is the incus. Or anvil. And then the third bone is another one that attaches to a membrane on this surface. It has a flat plate, and then extending from that flat plate is a kind of a handle. So it reminded individuals of a stirrup you would put your foot in if you were riding a horse. And so this is known as the stapes, or stirrup. So the purpose of these three sets, these three bones, is to amplify the sound waves. So the sound waves have energy. It's a radiant form of energy, and as they move through, they cause the primary tympanic membrane to vibrate, and then the malleus vibrates, and the incus vibrates, and the anvil vibrates. There are a couple of muscles that regulate this vibration. One stabilizes the stirrups, and the other one stabilizes the malleus. I'm going to write the names up there, but you're not responsible for them. You might pick another class that has them, but so the pedius stabilizes the stirrups, and tensor tympani, because of its association with the bone on the tympanic membrane, actually comes off a little lower here, but that's going. We have some regulations. So if the sound is too loud, or the amplitude, they're listening to somebody blowing leaves or something, these will tighten and dampen, kind of dampen that depression. Okay. So this space here has air. Middle ear has air, and that is available to the external environment because of the opening of the auditory canal in the nasopharynx. The nasopharynx, as we'll see in our digestive system, is that throat space directly behind the nose. So we're more susceptible to changing in pressures when we descend rather than ascend. But most of you have seen what happens to a partially empty plastic water bottle. As you descend from Lake Tahoe area, it gets all crumpled, all right? And that's because the density of the air molecules becomes higher and higher as you move closer to the center of the earth. Gravity's, those air masses, the nitrogen and oxygen and carbon dioxide molecules have a mass, and gravity pulls on them, so there's more of them, there's more mass as we move from higher elevation to lower elevation, and that creates a higher pressure. So as we descend in an airplane, or if we descend driving down the road, that pressure is going to move those air molecules closer together, and we're going to feel higher pressure creeping in on the external, from the external auditory canal on the primary tympanic membrane. The air molecules inside the middle ear are going to be at a lower pressure. They were equilibrated with the higher space that you were in the plane or in the, up in the mountains. And so what do you do to try to balance that out? Chew, stretch your jaw, and that helps to open up this internal auditory tube with muscles pulling on the opening in the throat and allow air in your mouth to flow up into balance this space. Okay. When my daughter was really little, she still has some problem with balancing and equilibrating that. I learned of a trick when you're descending in the plane. I would get 2 empty glasses with ice and put them over her ear. And that cold helps to condense the air pressure on the inside of the middle ear. She was too little really to give a bottle or something. You could just smell it after a while. They refused to do that. So there's just another tricks to do. So we have control of some of the magnification or amplification of the vibration in the middle ear. Notice we don't have nerve endings in here. We're not detecting sound, all right? There are nerves that pass through the middle ear. The facial nerve on its way out the stylomastoid foramen to get to the muscles of facial expression will pass through there, but there aren't any nerves for detection of either balance or of hearing. Those structures come from the inner ear. And the inner ear is another bony cavity. Give myself a little bit of space here. Within the bone, that temporal ridge of the temporal bone, and it is kind of carved out here in the bone. And we have 2 components now because of the inner ear being balanced and hearing. So this is, the inner ear is formed of a bony space that is then filled with a membrane tube, kind of like a wheel on a tire, bicycle, or the car that has an inner tube that has the actual fluid air in it. This is 2 fluid-filled spaces. So one structure that we have associated with hearing is known as the cochlea. That's a long coiled tube. And then for balance, we have 2 structures. We have semicircular canals, and another structure, the vestibule, formed of 2 spaces known as the utricle and the sacral. Okay. So we'll look at the structure for hearing first, and this is looks like a shell from which we get the name cochlea. And I'm going to take this, slide this diagram over, and take that cochlea and expand it. The stapes rest on a membrane that covers this cochlea. And then there is another membranous enclosure just inferior to that. Okay. We'll talk about it, I'll diagram it in a moment, but we have 2 membranes that allow the dissipation of this fluid that we find in the ear. I'm going to move this over to the side and redraw the cochlea. I'm going to kind of uncoil it slightly, make it a bit shorter. So here is the membrane that was closed by the stapes, and I'm just going to leave the stapes off for a moment. This membrane is identified as the oval window. This membrane is identified as the secondary tympanic membrane, or the round window. Round window is what you have in your lab hand. So what I've drawn is a bony space. Within that bony space is a membranous sac divided into 3 chambers. The fluid surrounding that is known as hairy lymph. So we're going to have hairy lymph down here as well. independent membrane is known as endolith. This is divided into three chambers, which I will draw in cross-section for you. The upper chamber is known as the scala vestibuli. The lower chamber as the scala tympani, and the middle chamber as the scala media. And this is coiled, right? Kind of like when you drive around a parking structure, you go around and around and around to the upper surfaces. So if we look at this in cross-section, the structure where we actually have our hearing receptors is along this area here. And I'm going to diagram that in cross-section and then label those membranes, because I've just shown you the spaces here. So we're going to take one of those coiled ramps. So that would be what I drew there in black. Scala vestibuli, scala tympani, scala media. And I'll throw up a picture on the screen in just a moment. So again, that's a cross-section here with perilip and those fluids. So this membrane is known as the vestibular membrane that separates scala vestibuli from scala media. Separating scala tympani from scala media, we have the base of the membrane. And so those two structures form the support for the scala media, also known as the cochlear duct. Resting on the basilar membrane are the sense receptors for changes in sound waves. And we have a series of supporting cells and cells with very long stereocilia. I'm just going to draw them plainly on here, but in fact there are several rows and they are in different levels. So if we were to take those cells right there, there are a taller in the middle, and there are 3 rows that create a chevron. I'll show you an image on the overhead in just a moment. And then that brings us to a third membrane that does not extend all the way across. But is found just over those hair cells. Okay. And this third membrane is known as the tectorial membrane. These are sound receptors, right? They're receptors, they're mechanoreceptors. So they create an electrical current not when chemicals bind to them, not when a flow of charged particles flow by, but when those stereocilia are actually bent, that opens up ion channels. And from these receptors, the nerve fibers extend back to the middle of our cochlea and become part of the vestibulocochlear nerve. So what happens is, as the sound vibrations occur, and they pass through the external auditory canal, cause the primary tympanic membrane to vibrate. That vibrates the malleus, which vibrates the incus, which vibrates the stapes. And now the stapes is resting on this fluid-filled space. on the oval membrane, and that causes the oval membrane to oscillate at the various frequencies of the sound. That sets up a fluid wave. So we've moved from a sound wave to a fluid wave. And as that fluid wave passes through the components of the ear, it, depending on its frequency, is going to eventually pass down and up on the basilar membrane, and it's going to push the basilar membrane up against that tectorial membrane. So the energy up here then comes up, pushes this tectorial membrane up against the basilar membrane, up against the tectorial membrane, and that causes those stereocilia, which are not real true cilia, they're long microvilli, to bend. And that creates the electrical current. So what we tend to see is the high frequency waves will activate sound receptors down here at the base. Low frequency waves will activate sound receptors up here at the tip. So high sounds here, low sounds will travel farther along the cochlea before they activate. Now there's a whole lot more to that because we hear more than just a sound. All right, we hear more than just a high C. You can recognize a person's voice singing the high C. You can tell if it's a clarinet or a trumpet playing. So there's a lot of other characteristics that come into play than just the actual frequency, and that has to do with our patterning, location along the tectorial membrane, the basilar membrane, which of these stereocilia are bent in which order, and so on. Now, I usually tell the story here of a mom on my son's soccer team. We were talking one day, and she was talking about how she had been cleaning her ear with a Q-tip. And the rule is you never put anything in your ear smaller than your elbow. And Q-tips are to be used to clear the external auricle here, but not going within the external auditory canal. And I break that rule almost every day. But she was using the Johnson & Johnson brand, not the cardboard ones, but the clear plastic that come in blue and green and pink. And it cut through, it slipped. She didn't bang her elbow or anything, and it cut right through the primary tympanic membrane and went into her round window. And she just left it there and went straight to the doctors. And so, being the anatomist and physiologist, I said, let me guess, you have hearing loss. And she said, yes. And I said, high frequency? And she said, yeah, because that's what the first things that would be damaged here by that Q-tip going through. And she said, but you know, I heard way too many high-frequency sounds anyway, so I'm okay. They were always irritating. You guys heard of the mosquito tone? The ringtone? Apparently there was a shop owner in England who, his shop tended towards older customers, but it was attractive on the outside to, not really hoodlums, but a lot of teenagers that would just hang around, and they were loud and smoking, and they would discourage his actual customers. And so he started playing just outside a very high frequency noise that sounds like a mosquito buzz. And once you get to the age of 23, 24, 25, 26, you're no longer sensitive to that high frequency. So it wouldn't even, the older customers wouldn't even hear the noise, but it was irritating enough for the younger folk, less than 25, to drive them away. So if you look on your phone, if you have smartphones, and look up mosquito ringtone, and test whether you're hearing. My son was like, really, Mom? So he played it in the car. He goes, you can't hear that? And I said, nope. He and my daughter could hear it, but my youngest son, who, because of his jaw damage, doesn't have that high frequency, his regular hearing is fine, but he and I were the only ones that couldn't hear it. So you can test your age and hearing sensitivity to that aspect. All right, so I want you to recognize cochlea for hearing. Recognize that this is a fluid-filled space, so we're transmitting radiant energy to fluid wave energy to mechanical energy as we change the structure of those hearing components. And that's where we get the cochlear portion of our vestibulocochlear nerve. All right? Now, let me pull the screen down to illustrate those structures as well as to move on to the second part of our apparatus, which is the balance portion. Yes?
I read that if there's pressure on the oval window, then the round window will like decrease or relieve pressure.
Well, the purpose of the round window is, so if you slap your hand against an aquarium, okay, create that wave motion, the energy has to be dissipated. And so the round window is into the middle ear cavity is how that pressure wave in the fluid, then that energy is dissipated rather than going into your skull, bone, and all of that. So the wave travels back out to the round window, and then that air fluid into that pressure back to the middle ear, the air of the middle ear cavity. All right, so this is showing a cross section through the cochlea, so you can see it looks a bit like a chambered nautilus. And so this would be one coil, cross-section through a coil with the scala tympani and scala vestibuli, and then this would be the scala media, or actual cochlear duct, and a higher view of that. So that's kind of what I drew earlier on the board. And then this would be our tectorial, our basilar membrane, or our tectorial membrane, and then the specialized hair cells. And here's another view of those. And then here's a scanning electron micrograph, colored by computer. They are not actually yellow, showing the chevron shape. And then there are actually 3 rows of them, okay, that are shown in dimension. And so stereocilia, meaning they're tall like cilia are, but they're actually microvilli, so they don't move. All right, they have the actin components in them. So if we go back to then our structure of the inner ear, this is where we were looking with the round window associated with scala vestibuli and the oval window with the scala vestibuli and the round window with the scala tympani. And then that perilymph is continuous with this larger space here. All right, so that is what we see associated with equilibrium. There are 2 types of equilibrium. There is dynamic and there is static. So we're going to be looking at the semicircular canals for dynamic and the utricle and sacral for static. So in the semicircular canals, let me pull this back down again. So we have, in the semicircular canals, we have 3 ring-like structures. Again, a bony channel, and then a fluid, like an inner membrane, okay, a fluid-filled space. And we'll look at these semicircular canals in lab as well. So the semicircular canals have the same endolymph and perilymph component. And at the base of each semicircular canal is a dilated region that contains a gelatinous material, and again, more stereocilia. And this gelatinous material can be bent as the fluid is circulating. So the 3 semicircular canals are at right angles to each other. And we have an anterior, posterior, and superior that we'll label in lecture. But as the fluid is circulating through, depending on the tilt of the head, all right, they'll affect one semicircular canal more than the other. And so semicircular canals are effective in dynamic equilibrium. So when I spun last Wednesday, and then stopped and looked at you guys, that was the semicircular canals that were being affected. So there's inertia first. As I start to spin, my head is turning, but the fluid in the semicircular canals is not moving yet. Kind of like taking a glass with a piece of ice in it and starting to swirl the glass. The glass is moving, but the ice is stationary for a while. And then eventually the fluid in the glass is moving at the same speed and direction as the glass, and now when I stop the glass, that fluid continues to spin. So the same thing happens with the semicircular canals. When I start to spin, that fluid is not moving, it eventually catches up, but when I stop spinning, that fluid continues to move and sends that message to the cerebellum, even though my eyes are saying, nope, I'm not turning anymore. Okay, so I want you to recognize the semicircular canals for dynamic or moving equilibrium. Then within this space, the vestibule, we have the utricle and saccule, which are two connected rooms. They also have hair cells with stereocilia embedded in a gelatinous material. But on the top of this gelatinous material are small mineral crystals called ear stones. It's kind of like, back to my favorite biological material, Jell-O. If you were to make a 3-foot tall mold of cherry Jell-O that you fill upside down, all right, and you put the cherries at the bottom, and then when that Jell-O hardens, you turn it upside down and take off the mold. It's going to be top heavy because you're going to have all, these cherries at the top. So any slight movement is going to be magnified by the weight of the cherries at the top of that gelatinous material. And that's what happens with the otoliths. They magnify any tilt of the head. And so utricle and saccule allow me to know if I'm standing, along with my proprioception, if I'm standing on one leg, all right, and I start to tilt over, that is exaggerated by those otoliths. And so the utricle and saccule are involved in what we call static equilibrium. So rather than the turning of the head, just the bent of the head from one side or forward or back. I'll just show you an image of those structures. It's kind of unique, and then we'll move to the eye. Showing the inner ear with the cochlea on the lower right. And then here we have the semicircular canal. Okay, so there are 3, and here where their sensory nerve endings are located. And then we have the utricle and saccule. So here are the sensory stereocilia embedded in just this gelatinous component for the semicircular canals. And then when we look at the macula for the static equilibrium, this is a blow up of those little otoliths or ear crystals that, depending on the position of the head, will fire more rapidly or less rapidly. The vestibulocochlear, cranial nerve 8, vestibule for balance. Either dynamic or static, so you should be able to recognize which structure, semicircular canal or utricle or saccule for which, and then the cochlea for hearing. All right, so let's move back now, and that would be cranial nerve 8. Okay, cranial nerve 2 is our special senses for vision. Half of the cranial nerve, of the 12 cranial nerves, have to do with the eye. 12 have to do with the eye. So I'll be identifying them as we go through. But first of all, we have the actual special senses carrying the electrical current activated by the photoreceptors with the energy of photons, light rays. So that's the optic nerve. We have 3 cranial nerves that have to do with the innervation of external muscles that move the eyeball. So we have ocular motor doing 4 of the 6, and trochlear and abducens doing 1 each. And then we have 2 other nerves that have to do with the eye. What nerve is going to carry pain, touch, and temperature? Not that we feel temperature that much, but pain and touch from the eye. Not facial. That's the other nerve, but not for pain and touch. Trigeminal. That's our sensory nerve from the face. All right. So you get a scratch on your eyeball or pain, whatever. That's going to be trigeminal, the first branch of trigeminal. And then facial is the other nerve. What is facial going to do that has to involve the eye? It's the autonomic or parasympathetic component of facial. Tears. Okay, so those are the 6 cranial nerves that have to do with the eye. Now before we look at the actual structure of the eye, we have some accessory components. So our eyelids, levator, remember the muscle that closed the eyelid was the auricularis oculi. All right, we also have a muscle we'll talk about in a moment that opens the eyelid, called palpebra. The 2 eyelids meet on either corner to form the commissures. And then on the needle aspects of the eye, we have the peduncles, the openings for the nasolacrimal ducts to drain the tears. Okay, I'm going to leave this diagram. We'll come back to, well, before I leave that, let me just show these structures here, the accessory structures of the deep surface. So here the auricularis oculi has been removed. You can see the lacrimal gland, which is in the upper outer component. And then I'll diagram the eyeball muscles in a moment, but I want you to note this one right here. This is auricularis oculi innervated by facial nerve. This is levator palpebrae superioris. So it lifts the eyelid. This is the only muscle of facial expression that's not innervated by the facial nerve. So we close the eyelid with the facial nerve, but we open the eyelid with the ocular motor nerve. All right, so it's going to open the eyelid. All the other skeletal muscles innervated by ocular motor have to move the eyeball itself. The levator palpebrae superioris pulls that upper eyelid open, levator palpebrae superioris. I'll show it to you in lab. We only have it on one of our charts. All right, so you just need to make a point of noticing whether it's going to the eyeball or going to the eyelid, so that you're not confused by that component. All right, I'm going to turn the lights back off and diagram on the board here for a moment. This is the upper eyelid. This is the superior and inferior. And the cornea probably doesn't bulge out quite that much, but I'll go ahead and leave it that way. We have a serous membrane that is attached to the inner aspect of the eyelid. And then to the upper, goes around the eyeball itself, over the cornea. Anybody ever, I used to wear hard contacts and I was always losing them. I think I'd be looking all over the carpet in my dorm room and it'd be in the back of my eyeball. It can't really go all the way back because we have this membrane that covers it, but you're sitting there, practically pulling your eyeball out, trying to squeeze it around so it comes back down. But this membrane is known as the conjunctiva. Ever heard of conjunctivitis or pink eye? Pink eye is just an infection or inflammation of the conjunctiva. Where it's attached to the eyeball itself is identified as the bulbar, or ball, conjunctiva, and where it's attached to the eyelid, it's the palpebral. So that's just a serous membrane that covers those components. And then we would have the, kind of tucked away up here at the top, we're going to have our skeletal muscle that lifts the eyelid, our levator, palpebral, superiorus. Now I'm going to leave that up there because I'm going to draw some cross sections in a bit of the eye. But when we look at the frontal view of the eye, let me bring that over here. Tucked up on the upper outer wall of the orbit, remember we have our frontal bone and our zygomatic bone and structures up there. We have the lacrimal gland. And that's innervated by cranial nerve 7 or facial nerve. And from there, ducts pass through the wall of the orbit to release tears onto the surface of the eyeball. If we had a drainage duct right here on the lateral aspect of the eye, the medial aspect of the eye would be dry. Okay? Or if we had it up here, it would be difficult to drain from the root. So our drainage is diagonally across the eye. And then the opening is in the medial aspect of the eye. So we have now in the commissure area and the peduncle of the eye, we have this drainage collection. So we have the openings, the punctum, if you will, for the openings of the lacrimal sac. And then that narrows to drain into the nasal cavity as a nasal lacrimal duct. And that opens up behind one of the conchae, the nasal concha. So you've probably seen videos where someone's taken a piece of corn or a little tiny snake and passed it through that duct and it comes out their nose. Mine aren't like that. I could see if mine are wide enough to try to do that. Okay, but these are still also considered accessory structures of the eye. The tears help to maintain the surface of the eye. It tends to be rather dry and can be scratchy if the tears aren't produced. I used to wear extended wear contacts, so I'd wear them for 3 or 4 or 5 months at a time. And then one night in October of 2004, when my contacts, I've been in there for a while, my eyes would start to get scratchy and I'd just take them out and throw them away and the next morning put in a new one. My eyes were scratchy and I took them out, and for, till last month, I wasn't able to wear extended wear contacts again because it'd be overnight, but my eyes just felt really dry. And it can be hormonal changes, it can be an inflammation, and so you've heard the Restasis ads on TV for ear eye drops and stuff. But starting about 3 years ago, I felt that my eyes were wet again. I wasn't putting eye drops in them anyway, but I felt I could wear my contacts overnight. And so I asked my eye doctor, I want to try extended wear again. I was wearing dailies. And he said, no, I'd already had. The reason I went to extended wear was I got an anaerobic eye infection when I was teaching a marine biology course in the Bahamas, and all we had were sulfa drugs. Try scuba diving with one contact in and one contact out. You have no depth perception. And so we got back to the States, and I was driving cross country to see my fiance here in California, and so I drove the entire distance without my contacts. I could see the exit sign by the time I got to it, so I knew how to get there. And I spent that first week in his apartment with a washcloth over my eye and sunglasses over that. My eye was so sensitive to light. And when I finally saw an ophthalmologist, he said, you've got an ulceration on the edge of your cornea, not in line with your vision. He said, but I don't want you to be taking your contacts in and out. So I thought he was going to tell me to just not wear contacts. And that's when he put me on extended wear. So I did extended wear from about 1988 until 2004. And I wasn't really good about it because I have eyes that are not very sensitive. And so a lot of damage can be done to my eyes before I recognize it. And I would wear them, instead of 1 month, I'd wear them for 3 or 4 or 5. And as we'll see in a few minutes, typically the cornea of the eye doesn't have blood vessels. It gets its oxygen from the air, okay? And so when I would cover that up with contacts, as the contacts would get a proteinaceous covering on them, then I go to see the eye doctor for a checkup, and they could see 1 to 1.5 millimeters of blood growth into the cornea. And they go, you've not been taking your contacts out. And then shortly after my eyes got dry, I developed a couple of other ulcers, a little closer to the midline. So my ophthalmologist is like, no, you've got a history now of 2 anaerobic infections, meaning the bacteria like it where there isn't much oxygen. And he said, I don't want to risk that again. So naughty me, I went to a different optometrist a couple weeks ago. Paid out of pocket and got extended wear. He was just like, I know I can wear them again. So I wore them starting spring break. I wore them for 2 weeks and just took them out for the first time there for a month. But I took them out Friday night. And it was just starting to feel scratchy. So I've been wearing dailies for the last couple of days. And we'll see. We'll try again with I'm not going to wear them for 5 months at a time. So when we look at the structure now of the eyeball, we're going to look at the layers of the structure of the eyeball and then what specific components are present in each of those layers. Let me just show you some slides on the muscle. That's better than my drawing, then we'll look at the actual structure of the eyeball. So we're moving to the 6 muscles that move the eyeball. These are considered to be extrinsic eye muscles. Meaning they're outside the eye, they're skeletal muscles, they're voluntary. And then we have 3 sets of intrinsic or smooth muscles of the eye. So I'm going to go through their functions. They're pretty basic, and then give you a bit of a memory tool to remember which are innervated by which. Okay, we went over their functions real briefly last week with cranial nerves, but we'll look at them again. We did not cover these test-wise with the first unit on muscles. So there are 4 that form a framework. If you think of the eyeball as a block of, square block or cube instead of round, we have 4 muscles that are identified as rectus. You guys remember what rectus means? No? The rectus abdominis? Straight. Okay? So these 4 muscles are straight muscles that come along the long axis of the muscle. So from the top, we have the superior rectus. On the bottom, the inferior rectus. The medial aspect next to the nose, we have the medial rectus, and then we have the lateral rectus. Right? So those are the straight eyeballs, and their functions are to do what you would think. So coming from the top, the superior rectus is going to pull the eyeball up, looking up to the sky. Okay? So just looking straight up, superior rectus. From the bottom, the inferior rectus are going to pull the eyeballs down, so looking down to the ground. The medial rectus are going to converge the eyeballs, so going cross-eyed. All right? And then the lateral rectus is going to turn the eyeball outward, or diverge. Now, most of us can't look sideways at the same time. All right? As we move the lateral rectus, the medial rectus of the other eye is going to contract so the eyes track together. So those are the rectus muscles. And then we have two muscles that come in at an angle. The most angular one is the superior oblique. So here it is coming through the pulley I mentioned last week. And then it comes back at an angle to attach at the back of the eyeball. So even though it's coming from the top, because it goes so far back at an angle, it's going to turn the eyeball down, okay, rather than up, because it attaches on the almost underneath surface of the eyeball. So superior oblique is going to turn the eyeball out and down. So I am just barely seeing my fingers moving out here, okay? So superior oblique is looking out and downward, sometimes called the cheating muscle. If you look over at your seatmate's page. Inferior oblique does not go at an angle, but attaches from the medial surface of the orbit and comes underneath inferiorly. So it's a horizontal. So the superior oblique is this kind of check mark shape, inferior oblique coming at an angle. It also turns the eyeball outward but up. So looking up here, now I can see my fingers. That's I'm also using my lateral rectus and superior rectus. Okay. But if those were paralyzed, I could do that movement with my inferior oblique. So superior oblique is down and out, inferior oblique is up and out. All right, now, to remember those innervations, there's a little bit of a chemical formula. So superior oblique is cranial nerve 4. I find it easier to remember that superior oblique is passing through a trochlea or pulley, therefore it's innervated by the trochlear nerve. But if this helps you get to that point, that's fine. Lateral rectus is innervated by cranial nerve 6. Again, I find it easier to remember that lateral rectus is abducting the eye, moving it away from the midline, therefore it's innervated by the abducens nerve. Okay? But this helps, and then all the rest are innervated by cranial nerve 3 or the oculomotor nerve.
So what makes you go like cross-eyed?
What makes you cross-eyed? How can you do it, or just people that are cross-eyed?
Both. Like what?
Okay, well, muscle medial rectus. So medial rectus is going to converge the eyes. So if I'm looking at my finger, I go cross-eyed, that's the pair of medial rectus moving. The lateral rectus do not do that. Lazy eye can sometimes be due to some problem with the innervation of one of the eyeball muscles, or if for some reason the eye is patched and those muscles aren't working for a while, they have to be re-exercised. Okay. Now let's look at the structure of the eye. And the outer, I'm going to go through briefly here and then we'll resort back to slides. The outer portion of the eyeball is formed of dense, regular connective tissue. That part of the eyeball belongs to a layer, there are 3 layers, belongs to a layer known as the fibrous coat. It's formed of the sclera, which is dense, irregular connective tissue. It helps to hold the shape of the eyeball. Because if the shape of the eyeball is off, then our focal length can be affected. So this would be the sclera. And then starting about here and here, we have the cornea. The cornea is dense, regular. Light has to be able to pass through it. So it forms, if you think of the ligament, that's dense, regular. Light doesn't pass through a ligament, all right? But this dense, regular connective tissue is laid in layers. It almost looks like lasagna, and how you would take cooked lasagna and lay it at perpendicular angles. And it creates a latticework-like structure, like a crystal, and light can pass through that. Okay, the sclera is vascular. Anybody that's had really red eyes knows that the white portion of the eye is vascular. The cornea is supposed to be avascular. Okay? Yes?
Some people have like a thin cornea,
Mm-hmm.
and then how does that?
It's really not known how that is affected. I actually developed keratoconus about 4 months after my dry eye developed. In keratoconus with a thin cornea, what happens is the layers of collagen fibers are thinned, and the cornea actually can develop kind of a pimple or a bump. It's a form of astigmatism, any irregular surface of the cornea. So if the cornea is wavy, that's astigmatism.
And they close the eyelid so that way they can grow back or something?
Well, no, with the keratoconus, there's usually, it's not known, it was thought to be rubbing, but that's not really true. So the cause really isn't known. Mine was temporary, which in all the literature I read, I couldn't wear my contacts anymore, my eyeglasses didn't, so I went to got three different opinions. And keratoconus is, you never get blind from it, but you can have such severe vision problems that you have difficulty reading or seeing. Mine actually was caused by a drug, I believe. I started taking a drug called Topamax for my migraines in January of 2005. I developed the keratoconus in March. And if you read the side effects, it'll say blurred vision, but it won't go any more than that. I didn't relate it to the keratoconus. I had to go to hard contacts that cost $1,200, and I could only wear them for a couple hours at a time, putting drops in all the time. And like I said, I have insensitive eyes, so it was really unusual. I also suffered Topamax, from the Topamax suffered hair loss. So when I only when I was washing my hair, I would notice when I ran my hands through it, I'd come up with about 200 hair follicles. I never went patchy balls, and I have pretty thick hair, so it wasn't noticeably thin, but I was like, I don't like this. And I associated that with the drug, and so I stopped taking the drug. It took 3 years for my hair loss to stop, but by September, I stopped in June. By September, my keratoconus was gone. The following year, I saw the ophthalmologist. They made a new prescription for new contacts. I never bothered to pick them up. The year after that, all the measurement, they'll measure the shape of the cornea. It was completely gone. And so I'm assuming that it was due to a side effect of the medication, how that affected the thickness. Usually the cornea is about 550 microns thick, and mine was 425 with the keratoconus, and I didn't have keratoconus, was like 375. So there was like a 25% change. But, so what you're thinking of where they close the eyelid must be some other cause I'm not aware of.
Yeah, it happened to a friend of mine, and then they closed the eyelid, so that way, like it would not do much damage. Because he has like very thin.
Did they use it? What treatment did they use? Did they use
just
something
closed
that's,
this,
but I mean, to increase the collagen production?
Nothing pretty much. I mean, just refresh AM/PM, just the medications, yeah.
So even with this, they don't know exactly what causes the loss of collagen. Okay, so the cornea with that plant of dense, regular connective tissue is transparent. It's going to be the first structure that a light passes through when we go through the light pathway. And then deep to the cornea, we have the vascular tunic. And this is where we have most of the function, not the retina, but other structures. So I'm going to draw this in red. We have a lot of blood vessels available, and it's also the structure that forms our intrinsic muscles. So this is going to be the vascular tunic. And it contains the choroid. It's not the only blood supply to the body. Actually, this is pretty thick. It contains what are identified as the ciliary body, and there are 3 structures of the ciliary body, and then finally the iris. And the iris has smooth muscle, has radial muscle, which when that constricts, dilates the pupil. Sorry, circular muscle. And then it has radial muscles. Let me erase this here, got that wrong. Constricts the pupil. And radial muscle, when that shortens, you can see that it gets, the space gets larger. So this would be to dilate the pupil. In our ciliary body, we have several structures. We have ciliary processes. And that secretes aqueous humor. We have ciliary muscle. And we have suspensory ligaments. So here would be the lens of the eye. It's a capsule made of layers and layers and layers of epithelium. Attached to that are suspensory ligaments. And then the ciliary muscle here adjusts the tension of the suspensory ligaments. I'll come back to that in just a moment, but I want to kind of show you, these would be the ciliary processes. So ciliary processes secrete aqueous humor. That's the water that we find, the serous fluid that we find between the lens and the eye. And ciliary muscles contract to loosen the suspensory ligaments, and that results in the lens rounding up. So let me just spend a few moments explaining that. Here is the lens, and the lens can be flat, flatter, or it can be round. When the lens is flat, it doesn't bend the light as much as it does when the lens is round. When the lens is round, we use that to look at things close up. Because there's a shorter distance to focus that light on my retina. When things are far away, you don't have to bend the light as much. All right. So when there's, yes?
So the it provides.
The Cori process?
Yeah, it provides to the eye, but like, does it also help the flow of CSF? Is that the same one?
Not the CSF. So this aqueous humor, and I'll come back to that in just a moment, okay? So don't let me forget that. So the lens are attached to collagen fibers in a circular fashion. These would be the suspensory ligaments. And the suspensory ligaments are attached to the ciliary body, which contains ciliary muscle. It's kind of a paradox here, but when the muscle contracts, the suspensory ligaments are loosened. So this is a sphincter-like muscle. Remember a drawstring-like muscle? So when that when the ciliary muscle contracts, it actually does this. It's a circular muscle. So when it does this, it comes closer to the lens. So what that does is it loosens up the ligaments. So the ligaments become become looser, and now the lens will round up. When the ciliary muscle relaxes, it moves farther away from the lens, and that tightens the ligaments. The lens becomes flat. All right? Contraction of the ciliary muscle is oculomotor nerve. So this is an intrinsic muscle that the oculomotor nerve innervates. Oculomotor nerve is for close-up viewing. Your pupil constricts to eliminate stray light waves. The ciliary muscle contracts to allow the lens to round up, and you can see things in focus at a close view, which is why with age, the lens is stiffer. It doesn't round up as much, and people start holding things farther and farther away to be able to have them in focus. All right, that has to do with the stiffness of the lens. Okay, back to your comment on the aqueous humor. The aqueous humor secreted from these ciliary processes passes through this space of the eye into this space of the eye, and it is reabsorbed at the area where the cornea and the sclera meet. It's not CSF, it's aqueous humor.
But the carotid plexus does flow of CSF.
It's part of the sinus component pathway that we'll talk about with the
So is it the
cardiovascular.
same thing?
That's outside. That's near the trigeminal nerve. That's outside the eyeball along the sella turcica.
Right, but is it like the same
No,
nerve,
it's-
or is it a different nerve?
No, it's different. It's a different pathway.
Okay.
I mean, this fluid comes from blood, but it's a different circle. This is going to go back into the venous sinuses, so it's going to go back into the internal jugular like that carotid plexus is, but it's prior to that.
So if this was on like the lab exam, would we have to put the carotid of the vascular tunic, or?
No.
Okay.
All right, so the last, we've got about a minute here, the spaces of the eyeball we'll cover in lab as far as chambers and posterior chambers, okay? But the last component of the eye is the nervous tunic. And that's going to be right here. And those are photoreceptors that send their ions, their axons or central processes out the optic nerve. Okay. At the optic nerve, there are no photoreceptors. So that is known as the blind spot. If you have your online lecture notes, there's an X and an O. Can I just borrow yours? So I can only close my left eye voluntarily. I have to use my finger on my right eye. But if you're looking with your right eye, you want to focus on the X. And when you bring it up to this part right here, now the O is disappeared. That's because the light waves on the O are landing on my optic disc, which there are no photoreceptors, and so there is no image produced. As I move it closer or farther away, they're going to land here or here, and then I will see an image. Okay? So these are where, remember the second location, we have bipolar neurons in our olfactory, and our bipolar neurons in our retina. And then the cells leaving the optic nerve are similar to the sensory neurons. Those are second-order neurons that go out there. So there are no photoreceptors here, and that's why we have the blind spot. Okay? So in lab, I'll talk about the chambers, or Dr. Lancaster, their structures that you'll identify in lab, and so we'll cover those components there.